Let's talk about some other proteins that are found on the surface of T cells. So we've already talked about the T cell receptor alpha and the T cell receptor beta protein, which are the two very important proteins that make up the T cell receptor. But it turns out there's more in this protein complex that's found on the cell surface. There are other proteins found next to the T cell receptor proteins called the Cd3 complex, and these are made of a number of different proteins. You don't really have to know their names, but we can refer to them as CD3 or the Cd3 complex, and they flank the T cell receptor alpha and beta proteins. And there's another protein in the middle that is bound to the cytoplasmic tail of T cell receptor alpha and beta proteins, and it's called the Zeta proteins. So what are the functions of these proteins? Well, first let's give this whole thing a name. We're going to call this the T cell receptor complex. And we'll talk about this in context when T cells become activated. When we talk about the T cell receptors, we're typically just talking about the alpha and the beta chains. But the whole complex is going to become very important when the T cell receptor becomes activated. So during T cell activation, something's going to happen to the cytoplasmic tails of these proteins. And you could probably think about what we observed in B cells when they activated. Same thing pretty much happens in T cells. We're going to have a kinase that's going to phosphorylate tyrosine residues in the cytoplasmic tails of these proteins, right? And those regions where we're going to find these tyrosines are going to be called intracellular tyrosine associated activation motifs. Intracellular tyrosine activation motifs. So this is just like in B cells. So when a T cell becomes activated, proteins get phosphorylated, a signal is sent into the nucleus, and we can activate the T cell. We'll go over this again in Chapter 8 when we show how T cells become activated, but we need to just talk now about the fact that there are these other proteins found hanging out with the T cell receptor, and they play a very important role in T cell activation, signaling into the cytoplasm, signal transduction to the nucleus. But we'll come back and visit those later. So there's two more proteins that can be found on the surface of T cells that we need to talk about what those proteins are and what their function is. Let's start with the CD8 protein complex. So if the CD8 proteins are found on the surface of the cell, we're going to call the cell a CD8 positive T cell. So some T cells in the body have CD8 on its surface. We call this CD8 positive, and that can help us tell the function of this T cell. If this T cell becomes activated, it will become a cytotoxic T cell and wander the body, killing infected cells. And we'll see the function of CD8 in a minute. Some T cells in the body don't have CD8 on their surface. They have CD4 on their surface. So if a T cell has CD4 on its surface, we call it a CD4 positive T cell. And that helps us identify the function of this T cell. If activated, it will become a helper T cell, which basically means it will release cytokines to help stimulate other parts of the immune response. So in your body right now, about a third of your T cells are CD8 positive, and about 2/3 are CD4 positive, and they're wandering the body looking for antigens to bind. Now, this brings us to the function of CD4 and CD8. What do they do? Well, this is involved in antigen recognition. So, from the introduction in Chapter 3, we talked about this concept that there are these antigen presentation proteins called MHC1 and MHC2. So here we're looking at a cell, this green cell, that has the MHC1 complex on its surface. And so the thing to know about the function of CD8 is that CD8 binds MHC1. So when a CD8 positive T cell is wandering the body looking for a peptide, that peptide is going to be loaded onto the peptide binding domain of the MHC1 complex. If a cell has MHC2, or the MHC2 protein complex on its surface, that cell will be examined by a CD4 positive naive T cell, because the CD4 protein binds MHC2, right? And so CD4 positive T cells wander the body looking at peptides that are loaded on MHC2 complexes. And so the easy way to remember which cells match with which MHC receptor is the number 8. The two numbers have to multiply to get 8. So if you look at CD8 binding MHC1, 8 times 1 is 8. If you look at CD4 cells binding MHC2, 4 times 2 is 8. So you can always keep track of which CD4 cell binds to which MHC. So a CD4 T cell is not going to look at the MHC1 complex, that would be 4 times 1, and CD8 is not going to look at MHC2, that would be 8 times 2, that would be 16. So you can always keep track of which MHC, 1 or 2, goes with which T cell, CD8 or CD4. So we'll talk about this more in the next video, but the reason that you have CD8 binding to MHC1 is because it brings this whole T cell receptor complex in proximity. And remember, the purpose of the T cell receptor, the function of the T cell receptor, is to look at these peptides that are loaded onto MHC1 and say, do you recognize this peptide? Does the T cell receptor recognize that peptide? If it doesn't, that naive T cell will go and look at another cell. But if it does, it will activate. And we'll again talk more about T cell activation in a later chapter. But if this T cell makes a T cell receptor that recognizes and binds that peptide very strongly, and T cell receptors should only be binding things that are foreign or non-self, that naive CD8 positive T cell will become an activated CD8 positive T cell and will undergo mitosis and mitosis. And we'll see in later chapters, when the T cell becomes activated, this T cell will wander the body, killing any cell presenting that peptide, because that peptide is foreign, so that cell must be infected by a virus, or it has as some sort of cytoplasmic pathogen. So the MHC1 complex allows CD8 positive T cells to look at the antigen being presented by those cells. On the other side, with CD4, CD4 protein complex is going to allow this CD4 helper T cell to look at the peptide being presented to it by MHC2, and the T cell receptor will swing up there and look and see if it can bind that peptide. If it doesn't bind the peptide, then the T cell moves on to the next cell and wanders the body looking for a very strong binding. If it finds it, if that T cell receptor has a antigen binding site that binds that peptide very strongly, that T cell receptor knows that that peptide is a foreign peptide. It's a non-self peptide. So that T cell, which is naive before, will become activated. And when it activates, when a helper T cell activates, it produces cytokines, as well as other molecules that are involved in cell-to-cell communication to activate the immune response. So you should know the function of all these proteins that we find on the surface of T cells, and especially with CD8 and CD4, their function in binding the MHC proteins. The next video will go into detail into antigen presentation and MHC complexes.